third webinar in the series organized by the Department of Biotechnology, Government of India, in association with DBT Wellcome Trust India Alliance. This series aims to highlight the role played by DBT, its autonomous institutes, and public sector undertakings in mitigating the COVID-19 pandemic. My name is Hina Latif. I work with DBT Wellcome Trust India Alliance, and I'll be your host for today's webinar. A little introduction about this webinar series. In the first webinar in this series, we had Dr. Renu Swaroop, Secretary DBT, Dr. Alka Sharma, Advisor DBT, and Dr. Kavita Singh, Mission Director, National Biopharma Mission BIRAC, as the experts to discuss DBT's multi-pronged strategy and action plan to address COVID-19. At the second webinar, which focused on how DBT is leveraging the startup and industry ecosystem to respond to the COVID-19 pandemic and how startups can be a part of the efforts, we had with us Dr. Manish Diwan from BIRAC, Dr. Tasleem Arif Sayed from CCAM Bangalore, and Dr. Jitendra Sharma, CEO of Andhra Pradesh MedTech Zone. DBT's autonomous institutes are actively involved in the country's fight against COVID-19 through the development of diagnostics, vaccines, and drugs. Some of the institutes are also training the medical professionals in collecting the samples. Since covering the vast work of all of the autonomous institutes in one webinar is impossible, in this webinar, we'll learn more about the efforts of three of these institutes: the National Institute of Biomedical Genomics, Center for DNA Fingerprinting and Diagnostics, and the National Center for Cell Science. To discuss more about the efforts of these institutes, we are joined by their directors, Dr. Saumitra Das, Director of National Institute of Biomedical Genomics, NIBMG, Dr. K. Thangaraj, Director of Center for DNA Fingerprinting and Diagnostics, CDFD, Hyderabad, and Dr. M. K. Bhatt, Director for National Center for Cell Science, Pune. Before we begin with the webinar, a few housekeeping announcements. This webinar is being recorded, and the recording will be available on the YouTube channel of India Alliance soon after the webinar. We would request you to share your questions with us by using the question box on the right-hand side of your screen in the console. These will be taken up by our speakers at the end of the three presentations today. Lastly, if you share about the webinar on social media, do consider tagging the Department of Biotechnology, Government of India, and India Alliance and the speakers. Use the hashtag DBT webinar on COVID-19 or DBT fights COVID-19. With this introduction, I invite our first speaker for the day, Dr. Saumitra Das, who's the Director of National Institute of Biomedical Genomics, to share his views with us. Over to you, sir.
Thanks to the organizer, DBT and India Alliance. I'm very happy to participate in this webinar, and particularly it so happened the other two speakers are also part of a bio cluster that DBT has formed between NIBMG, NCCS Pune, CDFD Hyderabad, and NIAB Hyderabad. And I could figure that this is the first time we are actually talking on the same subject, the COVID-19. So COVID-19 has done a lot of damage, but it is also, in a positive note, bringing in people together to address a challenge collectively. So this institute, National Institute of Biomedical Genomics, actually was started at 2009. And that's the time Professor Parthupratim Majumdar, the founder director, he established this institute, and with a vision that this institute will enhance the knowledge on human health and disease through genomics, and to translate the knowledge using appropriate technologies for promotions of well-being and improvement of genetics-based healthcare. So his vision at that time is now our mission to achieve. We are trying to collectively address, join hands and collectively address with all faculty members here to see how we can contribute to the human health using the biomedical genomics. Now, today's, as we rightly pointed out in the beginning, that this is actually the topic of the hour, the COVID pandemic, and this is a new challenge to us. But before getting into that, we have another two talks. I would like to introduce the organism. This coronavirus, particularly this SARS coronavirus 2 that we call as now COVID-19. Now, this particular virus is an RNA virus. In fact, this RNA viruses troubled us the most. It is true that we get much of the trouble from RNA virus, but it is also true that we have learned quite a bit of biology from this RNA virus. Now, this is the RNA viruses are the highly mutating viruses, and if you really look at this RNA viruses, they enter into the cells, and then by receptor mediated endocytosis, so is the SARS-CoV-2. The RNA then makes the protein. The protein is made into two polyproteins, the polyprotein 1A, and the second polyprotein is made by ribosome frame shifting. And then these proteins are used to make replication complex along with the positive strand RNA. From the positive strand RNA, the negative strand RNA is made, and then this negative strand RNA is used as a template to make more and more positive strands. But these are segmented RNAs, so these segmented RNAs make different proteins. Those proteins assembled again, and then the whole virus assembly actually is now covered with a smooth walled vesicles, and that's how they egress it, by hiding to the stiff challenge of the cellular environment. Now, as I mentioned, that the RNA viruses are very rapidly mutating, and partly because of its error-prone RNA-dependent RNA polymerase, which actually has a low fidelity. But these mutations also have some kind of a threshold, and it's actually self-limited. The 30 kilobase positive sense genome of the SARS coronavirus is kind of a trade-off between the genome length and the mutation rate. So the mutation rate compared to other viruses, fortunately, is not as much. But once this virus goes inside the cells, it actually hijacks the host machinery. There is a lot of RNA and protein interaction. It could be the viral RNA and the host protein. It could be the viral protein and the host RNA. There is protein-protein interaction. There are protein-lipid interaction. And all these viral and host proteins in the actually participates in the virus replication complexes. So the host factors, means like these RNA and the proteins or even the immune systems, are the key determinants of the viral pathology and the evolution, and many times that actually is the deciding factor for the severity of the disease in a particular individual. If we look at the viral genome for this SARS-CoV-2, it has an envelope, it has membrane protein, the spike protein, nucleocapsid protein, and as you see here, this is the genome. As I told you, it's a 5' capped RNA, and 3' there is a poly A tail like any other eukaryotic RNA. The genome codes for a single replicase gene, which actually occupies 2/3 of the genome. And the rest of it is actually, there are some structural proteins like the spike protein, envelope protein, membrane protein, and the nucleocapsid protein. The complete synthesis of the replicase polyprotein requires the ribosome frame shifting, as I mentioned. As you see, WRF1b can be synthesized only by the ribosomal frame shifting. And this N protein is the nucleocapsid protein, which binds to the genomic RNA to form the viral capsid. And the spike, envelope, and the matrix are the envelope proteins, wherein the S protein is the major envelope glycoprotein, which is involved in the fusion of the viral particle with the host cell membrane. Actually, to tell you the truth, I could figure it out that now all of you know much better than us, because I think of the media and because of the WhatsApp University, every information is actually available at your fingertips. Now, what actually happens for the RNA viruses are, when they are in the natural host, as I told you, because of the low fidelity of the replicase enzyme, the RNA-dependent RNA polymerase, often there is spontaneous mutations. And a lot of times there are antigenic drift, which we call, because of the mutation actually changes the epitope or the antigenicity of the surface proteins. But that is still possible to handle. But a lot of times, because of that, it ends up actually infecting some intermediate host. And in the intermediate host, when it undergoes further mutation, it might acquire something which is now, although it was not originally supposed to infect human host, it ends up infecting the human host. And that's precisely to some extent happened into this SARS-CoV-2 infection, and we thought that it has undergone an intermediate changes in an intermediate host. Now the spike protein actually interacts with the SCE2 receptor, and there are co-receptors contributions are also there. One of these is TMPRSS2, and both of them actually contributes. Now there are considerable debate going on that where from did we get this notorious virus. Now, if you really go by the genomic sequences and compare them, the phylogenetic tree suggests that we are actually this SARS coronavirus, human coronavirus, or this COVID-19, what we say it, is actually very close to the one that is in the bat and also from the pangolin. And if you really see, pangolin coronavirus is 91.02% identical with the SARS-CoV-2 at the whole genome level. But another thing that was pointed out in a publication, that 5 key amino acids of the for the SCE2 in the receptor binding domain of the spike protein. Now the spike protein is the one that interacts or docks into the SCE2 angiotensin inhibitor receptor to the cells, and now this SCE2 receptor interaction, there are 5 amino acids plays a key role, and these 5 key amino acids in the RNA receptor binding domain are consistent between pangolin and the SARS coronavirus 2 that infects human. And SARS coronavirus 2 contains a potential cleavage sites for the furin proteases. This is another observation which actually makes its more affinity or more tight binding with the SCE2 receptor in the human. So it looks like there is sufficient reason to believe that there is an interspecies transfer of this coronavirus, and the host factors plays an important role in deciding a productive viral infection. So as I told you that it's possible that it has come from bat, it has a direct contact into a pangolin, and from pangolin, because of a consumption in China, it might have slipped into human, and from human through droplets it's actually spreading to other humans. And you know, the droplet size is very important. If the droplet size is big, then it can contain more number of virus particles. If droplet size is small, it will have less number of viruses. So depending on how far you are, where from this droplet size, the bigger droplet or the smaller droplets infects the others, the virus load actually varies. But the dangerous thing is now it looks like there are some reports which suggest that it might cross the barrier, species barrier, and there are reports that there are tigers and cats are also getting infected. How they are infected and what are the host tropism, those things are actually not known. So at this stage, around in sometime in May, June, actually we conceived this idea that this is an idea by Secretary Dr. Renu Swarup and also our Honorable Minister Dr. Harshvardhanji, that why don't we just sequence 1,000 SARS-CoV-2 RNA using all these DBT institutes, and we later on make it as a pan-India, and we collaborated with other institutes as well. So primarily it was actually NIBMG was given the lead role along with ILS Bhubaneswar, CDFD Hyderabad, NCCS Pune, and InSTEM NCBS also helped in doing the sequences. And what we try to do it is we try to sequence 1,000 of this viral genome. Particularly NIBMG actually collected around 500 sequences RNA from different parts of India, and other institutes like ILS Bhubaneswar, CDFD, NCCS, and InSTEM NCBS, they have also sequenced in their facility and then contributed to this 1,000 genome. Now we have around 1,262 genomic RNA sequence for this particular virus, and we tried to find out the molecular phylogeny of the virus, which should indicate the track how the virus is evolving. And then the ultimate idea is to correlate this sequence variation in the viral RNA to disease severity and the transmission efficiency, ultimately leading to the identification of the strains which are associated with the enhanced pathogenicity. So what we did is essentially we have at each center we have collected naso or oropharyngeal swabs and collected the RNA, isolated RNA from there. those RNAs were actually transported, either sequenced on that facility if it is one of those primary centers, or like an NIBMG collected from ICMR, NICET, IPV GMER Kolkata, IISc Bangalore, AIMS Uttarakhand, then Maulana Azad Medical College in Delhi, THSTI Faridabad, Aurangabad, Wardha, Bhubaneswar. All these places we collected, and then Pune, they have collected from FMC Pune, BJMC, Biomedical College Pune, and all these places we sequenced it. And after we got all this 1262 sequence, we tried to make some sense out of that, and we tried to do a phylogenetic analysis, which we call a time tree. Now, in this time tree, we found that actually majority of these variations actually stopped after April. You can see that most of these places are now spreaded by A2A strain. Now, this A2A particular one is now is renamed or renomenclatured as 20A or 20B or 20C. Originally, it's all started with 19A from Wuhan, which is actually O type. There are some A2 and A3 type as well. And then there are some 19B, which is B1 and B4. But you can see that it started actually from March onwards, March and April, most of these divergence actually said. And then from May onwards, you can see it's swept out by the A2A strain all over India. If you really look at the pan-India picture, this is the situation. You can see that in March, you can see there were still some pockets. There are other types like O or B4 or A3, but then by June, you can see almost everywhere the A2A strain has actually covered it up. So the haplotype diversity is peaked between March to May, an early part of the outbreak. By June, A2A emerged as the predominant haplotype, and the temporal haplotype diversities landscape appears to be similar in India. So if you see that we have actually divided our sample collected from different parts like East, West Bengal and Orissa, South we collected from Karnataka and Telangana, West and West we have collected from Maharashtra, and North we have collected from Uttarakhand, Haryana and Delhi. If you see that all over by June, you can see A2A strain, which is with a signature mutations, are spreading and covering all the other, and then sweeping out the whole outbreak of this scenario. If you go by the haplotype distribution region wise, you can see within haplotypes there are different states appear to have different proportion. Like if you really see that we have actually collected from 10 different states. Northern India and Eastern India are kind of similar. You can see 20A and 20B showing a similar trend. But in the Western and the Southern India have a little different. You can see 20B is actually picking up more compared to the North and the East. East and North are similar, West and South are similar. Then we try to understand the viral lineage and how it is actually originated, which country it is originated or predicted to be originated. It looks like the 19A and 19B mostly came from China, and 20A, 20B, 20C, which we call A2A, from United Kingdom, Italy, and Saudi Arabia. There is 20A, also there are evidence came from Italy, Saudi Arabia, and in all Indian regions. But in case of Eastern India, 20A came additionally from UK and Switzerland. We got some actually in this Eastern India pockets from Switzerland as well. 20B was introduced mostly from the UK, and all regions additionally in Western India from Brazil, and Southern India introduced from Italy and the Greeks. So we can see that all over the world that there is a travel history, and if you trace the travel history, you can actually find out how they have contributed to the overall manifestation of these different strains in different parts of India. So we started doing research, and the first report was from Professor Partha Majumdar and Nidhan Biswas, who showed that SARS-CoV-2 belonging to this A2A type and possesses a non-synonymous variant D614G. Now everywhere you can understand these mutations all over in the newspaper now. This actually we reported from our institute as early as in May, and we showed that this we believe is the reason that A2A type enjoys a major advantage to infect and survive and has resultantly rapid swept all geographical regions. Then another study, actually I was part of that, is in June we have published that with a big group and only small number of sequences from West Bengal, and we showed that that A2A is the predominant clade in India, which is evolved in Europe, Italy and Spain mostly, and eventually replacing other clades in US. Initial samples from India was clade B1, which was closer to the original clade and the Wuhan clade, and subsequently
Dr.
one was
Saumitra,
mostly A3.
sorry
Now,
to
when we try
interrupt
to
you.
make sense out of these variations, we try to understand that D614G is so much. Is that possible? We can figure it out why this D614G is actually contributing so much. And then actually we showed that D614G is a part of the S1 crown, and this S1 and S2, although the receptor binding domain is in between, but the receptor binding domain might have an influence because of the mutation in the 614G. We also pointed out nucleocapsid mutations, and we showed that how that mutations can changes the disordered structure into a structured region and contribute to the virus assembly. We also pointed out how the mutations in the nucleocapsid can interfere with the microRNA binding sites. One of the study we showed that the 5' UTR can actually contribute to the replication or the translations of the virus, and looks like the strain that we have it in 2019 compared to 2003 what we had, there is a new stem loop appeared because of a mutation, and this particular mutation we are trying to see which host factor binds. One more study from our institute, they showed that how these mutations can predict, actually it's a predictive outcome, how it can alter the RNA binding protein, and the RNA binding protein will actually interfere with the microRNA binding. And all these publications actually gives an glimpse of what kind of studies are going on from our institute, so that we can heavily contribute that how the alterations in that genomic variations can affect the outcome of the disease severity. Now, to test it in a virus system, we are working on a virus-like protein particle. Now, VLP is the one which is non-infectious thing, and it doesn't have the nucleic acid to contaminate or infect. So with this, we are trying to have these mutations incorporated into the VLP and try to understand how this VLP with the mutations can affect the entry of the virus or the immunogenicity of the virus or the pathogenicity of the virus. We're using this construct, and this is a DBT sponsored and BIDAC sponsored project that we are trying to get it. So to conclude, I just wanted to highlight what is the findings is we have completed 1,000 genome sequencing across 10 states covering different zones, and it showed that A2A haplotype with this D614G mutation actually overpowered all the other mutations. And what we are going to do it is we are going to test this effect on this entry, immunogenicity, and the pathogenesis using the VLP model. We are interested to find out the host genetic polymorphism, how that confers susceptibility and protections from the viral infection. And finally, we would like to understand how different zone people are actually behaving differently to the disease severity, how much is the host genetic determinant is playing role, or the viral determinant like the viral mutations versus host determinant, the crosstalk can contribute to the disease severity. I would like to thank Professor Arindam Maitra from our NIBMG and the team, 1,000 genome team, my colleague, director colleagues, and all the collaborators in hospitals and institutes who helped in this pan-India 1,000 SARS COVID-2 RNA Genome Consortium, and of course my students in my lab at NIBMG and Indian Institute of Science, Bangalore. Thank you.
Thank you so much, sir. That was a very comprehensive presentation. I'm sure our audience has benefited a lot. We now move on to our next speaker for the day, Dr. K. Thangaraj, Director, Center for DNA Fingerprinting and Diagnostics, Hyderabad. Over to you, sir.
So good afternoon to all of you. I'm Thangaraj from Center for DNA Fingerprinting and Diagnostics. This institute is one of the DBT institute, basically to provide service and training in the area of DNA profiling and diagnostics of genetic disorders, and to carry out a basic and applied research in the area of modern biology. So this work which I'm going to present probably will fit with the mandate of this institute. CDFD is working on two different aspects, which I'm going to present now. One is diagnostics, other one is sequencing of virus genome. And also we have some future activity, which I'll come to the end of my presentation. So there are a lot of diagnostic options using antibody-based immunoassay technology, paper-based biomolecular sensors, whole RNA sequencing, CRISPR-based technology, and also there are several new technologies being invented by various laboratories. Particularly, I know that CSIR laboratories have found new technologies, but real-time PCR is the one which everybody uses. So we also used only the particular technology. The technology goes like this. We collect the sample. We get the sample, in fact, from a state government. That's a swab in viral transport media. And we isolate RNA, do cDNA synthesis, and use real-time PCR, and the report will go to the clinicians and they take further action on the patients. Basically, we collected the samples from almost all the districts of Telangana, and this is a layout of the COVID testing laboratory at CDFD. It's more complex. I'll not go into detail, but I'll show some of the pictures where CDFD staff and students have done the work, and this is where the place where those who have involved in the COVID testings with PPE equipment and performing virus inactivation in BSL-2 laboratory, and the DNA isolation from the inactivated virus sample, and finally setting up the real-time PCR. Altogether, we have analyzed 14,494 samples as on yesterday. Of that, 3,433 samples are positive. And I didn't put in this graph of August because we have only 20 days of data. Otherwise, it may show the down of the curve. As you can see, until May, both you can see the positivity is not that much. As the progress, the positivity rate has increased. If we stratified the data, male and female, and this is actually the general trend, males are very high in number compared to females, and the positivity rate percentage wise, if you look at, not much different. In fact, females are slightly higher and compared to male. This could be for different reason. And if you look at the age-wise distribution, you can see that the age between 15 and 40 are with high frequency. That could be because they are the one who moves out of the house even during the lockdown period. And in the case of more than 60, if you see the percentage of positivity, they are slightly higher. Otherwise, all other groups are almost the same. CDFD also involved in, other than just testing, CDFD also involved in validating the new methods which was invented by CCMB. The problem with existing method is it has to be transported in viral transport medium, and there's always a leakage is a problem. Therefore, this is hazardous. Whereas the new method was invented by CCMB allows the sample to be transport as such in the sense dry swab, and therefore it's easy to pack and unpack, and it requires very less time in RNA preparation. But this method also is very economical because there's no involvement of viral transport medium and RNA extraction. In fact, that method has been published yesterday by CCMB team. So we have tested few samples using this method, and this is the currently used method of RT-PCR using viral transport medium RNA isolation for the RT-PCR. And this is a new method. We have used 7 samples, and it's almost concordant in 5 samples. So, and even CT values are almost comparable using these two genes. There are only 2 discrepancy. That's not that one showed positive, other one is negative. On the other hand, there's inconclusive by old method, whereas the new method is positive. On the other hand, old method shows negative and new method is conclusive. And I think this method is waiting for ICMR approval. Hope that it would come soon. I'll now switch over to the sequencing of virus genome. This has been explained by Professor Das earlier, so I'll not go into detail of this. And we used 2 different technology. One is Oxford Nanopore, where we can get long read sequence using GridION. Other one is Illumina, where get shorter read of a sequence using MySeq. And this is a routine pipeline for analyzing the sequence data. We have sequenced 210 virus using Illumina 123 samples, Nanopore, Oxford Nanopore 95 samples, and 8 sample is common between these two to check what is the difference between these 2 technologies. After filtering, only 3 virus genome could not yield as expected the sequence, but whereas we got excellent sequence in 207 viral genome. As Professor Das mentioned, we have contributed 134 samples to Pan India project. When we try to look at the sequence of all the 207 virus genome, we found that there are large number of mutations which are at low frequency. Some are unique to our samples, and there are 18 mutations which are of more than 10% of the samples. More interestingly, there are 4 mutations which are shown with arrow marks are exclusively present only in our samples. That was not earlier been found in any of the previous samples, including Pan India samples, NCCS, ILS, and CCMB samples. This is very interesting. When we looked at the 18 mutations which are highly prevalent, majority of them showed that the asymptomatic individuals are having high frequency, and one particular mutation, C2T, at 6027 position, this is exclusively present only in asymptomatic. Of course, the sample size is very less for us to investigate further whether it gives some kind of resistance in expressing the disease phenotype or not. When we aligned all the sequences, and this is the total number of sequence, and these are the different genes, and this is the month-wise. Of course, it's not given gradient here, but overall scale is given here. These 5 mutations, which is shown here, are exclusively present in Wuhan strain, but that was only till month of early April. Beyond that, there was a new virus has been brought, as excellently explained by Dr. Das, that there are new virus strains where you can see they are present throughout the analysis period. We also seen there are some mutations which started appearing somewhere in the month of May or June, which is again disappearing from here. That could be because of different samples region in which we receive the sample. But these are very interesting. So some of these virus strains are distributed at least all over Telangana region. This has been already shown, but what we see here, that was in fact the pan-India, but this is exclusively from Telangana region, where you see that 20B, otherwise it's A2 in terms of previous classification, has almost 90% of our sample. So this 19A, so Wuhan strains, it's very, very less in frequency. 20A, and these are part of the A2 clade, is present in very, very high frequency. Another point which is very interesting here is, so this particular cluster, the samples have come from near Hyderabad, very close to Hyderabad airport, where we see very highly clustered and all of them are asymptomatic. Okay, so these are some of the points which we need to understand further what actually is there. So in the subsequent month, probably you can able to understand these in better way. Having all these things, I also thought that we should look at, so what is the total number of infection, what is the rate of mortality. I looked at the data till yesterday. You can see.
So sorry to interrupt you, but can we wrap it up in a minute or so?
I'll wind up.
Thank you.
So looking at the total number of infection, total number of death, but the rate of death which is given in the gray color is only 3%. So having population genetic background, I'm looking at the way in each state showing the data, that's the mortality data. I thought probably the genetic variation exists in large number of Indian population, where we have 4,635 populations, and each population maintaining the endogamy at least for the last 2,000 to 4,000 years, a quite different set of mutation. Therefore, that may have a bigger role in determining the disease infection, severity of the disease, all asymptomatic. So there are some example already from our own group where we have shown some of the genes and the mutation those genes are differing between severe malaria, mild malaria, and asymptomatic, particularly this low MBL level, what you see here. There are a few more examples. So we thought that it would be a good idea to sequence the whole genome of all the infected individuals that we have started in collaboration with the CCMB to understand the genomic variation and is responsible in causing the infection, resistant to the disease, or severity of the disease. Having BSL, animal BSL-3 facility in CDFD, that would allow us to do much more work in terms of culturing the pathogens, not only virus or other pathogens, using the animal model, screening for a drug, using these together is very, very important, and future it would give us much more option to do this kind of activity. So with that, I thank all the people who are involved, particularly our scientific team, Dr. Ashwin Dalal, Dr. Rashna Bandari, Dr. Murli Vashyam, Dr. Hari Narayan. They are the 4 scientists who are involved in that. And other than this, there are large number of CDFD scientific and non-scientific people, including large number of students whom we call as COVID warriors, are involved in that. With that, I thank for the opportunity to project what CDFD is doing for the pandemic. Thank you very much.
Thank you so much, sir. Another wonderful presentation for our audience today. We would now move to our third speaker for the day, Dr. N.K. Bhatt, Director, National Center for Cell Sciences, Pune. Over to you, sir.
So let me start by thanking DBT Wellcome Alliance and the organizers for giving an opportunity to put forth NCCS contribution towards present national need due to this pandemic. For the benefit of audience, I will start with a brief introduction about National Center for Cell Science, an autonomous institution of Department of Biotechnology, Government of India. Its basis was laid in 1986 as Cell Repository Program and subsequently was renamed as National Center for Cell Science, or NCCS as we know today. Over here, it has made its mark as one of the leading cell biology research institutes in the country and all over the world, being guided in its journey so far by Dr. Ullas Waq to begin with, subsequently taken forward by Padma Shri Dr. Gyan Chand Mishra, and then present DGCSR Dr. Shekhar Mandi. NCCS serves the nation by maintaining and distributing a large number of animal cell cultures to various colleges, universities, research institutions. By doing so, we encourage the pursuit of cell biology in the country. We also created a state-of-the-art infrastructure for the characterization and authentication of cell lines. The primary mandate of NCCS is to perform basic and fundamental research in various domains of cell biology, to serve as a national cell repository in country, and more importantly, to contribute towards building a pool of highly trained, motivated human resources by training and teaching. Next. The scientific domains which are primarily being worked to address various questions relevant to human health and disease are indicated in this slide. In addition to being a national cell repository, NCCS also established the DBT Center of Excellence National Center for Microbial Resources, as it's called NCMR, with a special mandate for the collection, identification, preservation, and distribution of microbial cultures. NCCS NCMR is the largest individual microbial collection center in the world. It has been instrumental in facilitating research in microbial ecology in India. To achieve this mandate and to carry forward scientific activities, we are supported by highly trained scientists and technical staff, PhD students and project students, and well supported by administrative manpower. Moving forward in the line with the theme of today's topic and the challenges being faced by all of us in the world, and presently more so in our country, is because of COVID-19 pandemic. Well, Dr. Saumitra has already given a bit detailed description about it. What I'll only highlight is the common symptoms of infection are fever, headache, muscle ache, cough, sore throat, and breathlessness. Looking at the timeline for the world to getting to know about COVID-19, how it has aggressively moved all over the world is shown in the slide. The first patient was identified with coronavirus symptoms was in Wuhan, China. As the virus started moving out of China to Thailand, WHO declared it as a global health emergency on January 30th, 2020. On the same day, first case in India was reported from Kerala. Subsequently, it was being detected in many countries, and as on March 11th, WHO declared COVID-19 as a pandemic. Presently, all over the world, more than 20 million cases have been reported from around 188 countries. 15 million people have recovered, and more than 7 lakh deaths have been reported all over the world. As for the spread of the disease in India is concerned, the timeline is shown in this slide. The first case being detected on January 30th in Kerala. It has been progressively covering all over India. As of 20th August, nearly 2.8 million cases have been reported, whereas approximately 6.5 lakh people are actively carrying this virus. Good news is that more than 20 lakh people have recovered. So far, India has recorded 53,000 deaths, which have been attributed to COVID-19 infection. It's likely that due to timely, effective control measures implemented by the central and state governments, the death rate has been contained to an extent. Presently, Maharashtra is one of the most affected states. It has more than 60 lakh cases reported, out of which 4 lakh have recovered. Currently, 1.6 lakh cases are active, with over 20,000 deaths reported. Pune, where NCCS is located, presently has more than 40,000 active cases. Now, having briefly mentioned about COVID and its spread, let's also look into consequences of being infected by this virus in brief. Following infection, the individual may be symptomatic or asymptomatic. This is likely to be dependent on many host factors as well as virus type. The host factors may play a key role. The host factors play a key role are immune status of an individual, severity of symptoms, factors such as age and existing comorbidities. By and large, it has been reported that children and young adults can be asymptomatic. Also, the mortality rate is affected by how prone is the individual to respiratory diseases, past history of pneumonia-like symptoms. Additionally, pre-existence of diabetes, obesity, hypertension, etc. have been related to the status of disease in individuals. As stated earlier by Dr. Thangarajan, primarily the testing of COVID is done by RT-PCR, and later on the tests where an antibody test can be done or antigen test can be done have been carried on. But what the newly added antigen test has been promoted all over, and what is happening is with antigen testing, however, in non-positive antigen tests, then a confirmation needs to be done by RT-PCR. Moving forward further towards the initiative taken by NCCS over the last 6 months are highlighted in this slide. NCCS have been actively engaged in various activities to facilitate the ongoing efforts against COVID-19 outbreak in the country. It is one of the government laboratories identified to carry out testing for COVID-19. Given its expertise in biotechnology research, NCCS confidently took responsibility because of its determination and commitment of the NCCS scientists, technical staff, and unconditional support by other staff members. Soon after it was approved as a diagnostic facility by DBT and ICMR and Maharashtra state government, NCCS carefully assessed the challenge ahead and made extensive and speedy preparations. These included reorganization of some of the research laboratories into diagnostic centers, procurement of supplies like PPE kits, formulating and validating a standard operating procedure, getting technical and scientific staff trained at the National Institute of Virology for COVID-related biosafety measures and sample testing. Registering the facility with the appropriate authorities, NCCS began testing samples for COVID-19 towards the end of April 2020. In addition to setting up testing facility, NCCS also initiated work towards biobanking of biological material, sequencing of viral genome, and COVID-19 related activities. The diagnostic facility at NCCS rose to its challenge by accelerating the rate of sample tested per day. With a determined team effort, NCCS has now tested over 17,000 samples. Further, NCCS has also provided guidance and assistance to institutes in Pune to set up COVID-19 testing facilities on their respective campuses, and also shared resources with neighboring institutions. In addition to testing, as already stated, a biobank of blood cells and plasma from infected and convalescent COVID patients has been created at NCCS, which will serve as an invaluable resource for future research as well. Until now, more than 100 samples have been stored. NCCS has also been facilitating COVID-related research at other organizations by supplying cell cultures which are necessary to carry out this work. Also, as Dr. Saumitra pointed out before, we have been part of COVID-19 consortium of DBT, so which was set up to sequence the genome of virus from clinical samples collected at different locations in the country. The initiative aimed at understanding the genetic variation of this virus across the country is being, as stated by Saumitra, by NIBMG, and involves national research laboratories. NCCS till now has submitted 19 whole genome sequences toward COVID genome and to the global database GAISED. Moving on from collective initiatives, next few slides will take you through the research initiatives which are being pursued by scientists at NCCS. This project is dealing with production of pseudotype SARS-CoV-2 in BSL-2 setting using VSP platform for candidate vaccine development and biomedical research use, which is being done in collaboration with IIT Indore through BIRAC funding. Presently, pseudoviruses have been established and testing for neutralization using standard antibodies is in progress. The efforts are also underway for generation of virus neutralizing human monoclonal antibodies against SARS-CoV-2 in partnership with IIT Indore, Genomics Technologies Private Limited, Bharat Biotech, and FMC, with support from CSR, New India Millennium Indian Technology Leadership Initiative. Sera from patients were screened for the presence of receptor binding domain specific antibodies. B cell from these patients which were positive for RBD specific antibodies were cultured. B cell clones were tested for specific antibodies against RBD peptide. Selection of positive clones secreting RBD specific antibodies is being done at present. Additionally, as a proof of concept, generation of vaccine candidate using a mix of synthetic antigens that can elicit SARS-CoV-2 IgA antibody response to protect lung and mucosal surface is being undertaken. In this project, antigenic peptides have been designed and synthesized. Mice have been immunized with synthetic antigen peptides. At present, sera has been evaluated for immune response by ELISA. And the fourth one being also that using machine learning tools, diverse viral sequences reported from different parts of the world have been analyzed, and 4 peptides have been identified, which have shown strong binding affinity against main protease of SARS-CoV-2. Simulation studies suggest that peptides are stable. Further extended computational studies are underway. And all this work described in this presentation was possible only because of support extended by NCCS staff, either for testing, biobanking, and genome sequencing, as well as research activities. Also, the support extended by Department of Biotechnology, Government of India, Indian Council of Medical Research, New Delhi, NIV, Pune, BGA Medical College, Pune, FMC, Pune, IDSP, and DMR for helping us in achieving our goals. And we'll continue to do that. Thank you very much.
Thank you very much, sir. We now move to the question and answers round for about 10 minutes. May I request all our speakers to switch on their cameras? Thank you, sir. So the first question that we have has been a very popular one throughout our COVID-19 webinars. It is, how do you compare amongst the efficacy of the RT-PCR test, antibody detection, and antigen-based detection for COVID-19? Open to you all.
Can I answer this question? Yeah. Sure. Now, RT-PCR test is a gold standard for detecting virus. As far as antigen testing is concerned, what has been reported is, though antigen testing is very efficient and rapid, but however, the non-positive patients from antigen testing have to be re-screened for by RT-PCR to confirm they are negative. So there appears to be something called percentage which is positive by antigen versus RT-PCR. There is a difference. So it appears that the people who are antigen test positive are okay. They are classified as COVID positive. However, the test which were negative do not assure that whether they are still infected or not. So there is a debate on this. Therefore, that's ongoing in the medical community. As far as antibody testing is concerned, it is basically, the person has probably recovered, and therefore antibody is present.
Thank you, sir.
And to add that, because recently when I was interacting with one of our clinical collaborators, say that the person who infected more than 2 weeks back still doesn't have the antibodies. It means there is something else, some other mechanisms exist. Therefore, we cannot really rely on antibody-based test.
Thank you, sir. The next question is for Dr. Das. How the 1,000 Genomes Project and Biorepository will have an immediate impact against the pandemic?
Well, if we get to know that how this virus is evolving, how quickly virus is evolving, See, you have a specific vaccines or a specific diagnostic or a treatment regime going on, but suddenly if the sequence changes, they might have some direct or indirect impact on those. So I think this kind of surveillance is always important to have the genome information, whether they are fast mutating or not, because there are some reports now from Malaysia that there is a spot of severity. Now, whether something happens in, so it's essentially a preparedness as well to counter future pandemic. And regarding the biorepository, it's always important because you can always go back and then check it what actually happened to that, and then you can address a lot of biology questions using those biorepository, which was not possible before, because all these genomics informations are association studies. But for validations, you need definitely the culture. And for that, if you can go back to that same patient, virus isolated from the same patient, and then test your hypothesis, then that's the much more confirmatory results.
In continuation to this, there is another question for you. Do you believe that a vaccine based on the spike protein will be effective for long? And what are your views on the mRNA vaccines coming up?
Actually, both of them are spike protein targeting the epitopes within the receptor binding domain of the spike protein. So one, the basis is the spike protein itself, the adenovirus based, and the other one is actually mRNA. But that will also make the ultimately the spike protein. Now, this is the, as I mentioned already, this is highly antigenic, and then that should actually, people have shown that it has the neutralizing capacity. I mean, the antibody raised against the spike has the ability to neutralize. So that has been tested. And it has been also showed that even if there are mutations in the spike protein, like this D614G, it looks like it is still the convalescent patient serum can neutralize that, suggesting this antibody that this spike protein is raising is good enough to neutralize, and it can tolerate one or two mutations here and there. So it's not going to affect the vaccines as a whole, as we are threatened.
Thank you, sir. The next question is for Dr. Thangaraj. What are the reasons for less quality reads from nanopore, and can nanopore be used for sequencing?
Yeah, nanopore gives advantage of sequencing a large fragment. Okay, if you do Illumina, it gives a shorter read sequence. Assembly will be a problem. So if you use both, actually, if there is some gap, it can be. So that's advantage of using both.
Thank you. The next question is for Dr. Bhatt. Have any new receptors been identified for COVID-19 apart from ACE2, considering the large spectrum of the symptoms caused by the virus?
Well, as of now, I'm not aware of it, but I certainly feel that down the lane, when we know more about this virus, there would be a possibility that we will find some other receptors which will facilitate that. So as of now, the attention has been focused on ACU2 only. So not much information is available.
A question which is open to you all is, are there any hotspots for mutation in the genome of the virus?
Oh, that's Saumitra.
Right. I mean, if you really see, there are hotspots in the 5' UTR and the 3' UTR of the virus, and there is already one mutations in 241 positions. And then I actually briefly showed that that is almost like 92% of the genome that we have sequenced so far. This mutation prevails. And similarly, in the 3' UTR, there are mutations which already reported. Other than the spike and the nucleocapsid, also we have reported there is two back-to-back mutations. I don't remember the number, nucleocapsid, and we have reported it also in publication, and that might affect the virus packaging. So and then that mutations actually independently NCBS and in STEM, they have also sequenced and then used a different filter for a cutoff. And they all in their screen also it came out that this is a hotspot for the mutations in the nucleocapsids. So there are actually already identified. And as Manoj also said, we don't know, there are so many. We really need to do deep analysis of the sequence that we have obtained to really make sense out of that and what all things is happening all over the world. There are a lot of data, but I think we need to have time to have proper analysis.
Thank you, sir. Dr. Bhatt has a question for you. Is there any scientific study to substantiate the claim that Indian population's immune system is more robust in the light of high recovery rates and low mortality in India compared to the rest of the world?
Well, again, I think I'm not the expert in immunology, so therefore I would not be able to answer the question appropriately. But having said that, obviously the healthy people sustain infection better than non-healthy people. So whether it is specifically linked to whether we have better immunity as such, I'm not sure whether I'll be able to answer that question, and I would not guess any more on that.
So I can try to answer, although I am not an immunology expert also. But what I'm just saying that the level of exposure for an Indian individuals compared to an American individuals, level of exposure to pathogens are definitely different. And the level of exposure definitely prime your immune system to counter this kind of pathogens in the future. So maybe that probably helped us. We don't know whether it's a strong or weak. It's essentially we are geared up to handle more dangerous pathogens than compared to the people who are living in the bubble.
Thank you, sir. One question to wrap up the webinar, which is open for you all, is what are some learnings for research in academic institutions that we can take from this pandemic for the future?
Research topic.
Yeah, so what are some learnings that we can implement in the future in research in academic institutions?
No. Okay. See, to deal with a pathogen of this level or this effectivity, obviously you should have facilities, which is paramount for anything with the virus. So therefore, the places which have BSL-3 facilities have capability of dealing with virus. As for other labs which work with BSL-3 facility for virus, or they are working on BSL-2 facility, I think it becomes more for us to do a work on cell-based or a plasmid-based studies. And I think these are ongoing, and therefore it's too early time. It's just been 6 months to understand the virus. So obviously, scientists will have to spend considerable amount of time either using through facility or BSL-2 facility or cell-based assays before we understand more about this virus. So therefore, what it is said is obviously not labs are actually prepared to use BSL-3 facility. And we had the similar challenge when we started BSL-2 testing with this testing. So I think if somebody has to dig deep into it using live virus, then BSL-3 facility is mandatory. But cell-based assays, if you derive on DNA-based work or plasmid-based work, cell culture work, that can be done in every lab. And I'm pretty sure down the lane in 6 months we'll have a lot of literature coming out on this.
So, and also, as an academic institute, what I feel for other diseases like non-communicable diseases, it's fine to work on different aspects. But particularly for communicable diseases, where such kind of pandemic arise suddenly from nowhere, it's also important for those who are working on infectious diseases or any other organism at infectious level to extend their research in terms of understanding host pathogen interaction and interact with hospitals to expand their research in terms of actually researching in infectious diseases, not only working at the laboratory level, expand it to hospital to understand both disease and also mechanism of infection and minimum.
Yeah, I would like to comment on the public awareness. I think from this COVID-19, we have actually learned how much it could be beneficial to have if you increase awareness about these things. And now people are all aware of it because of this wide publicity in the media and then WhatsApp and then other places. So that's one thing, and people knows how to protect themselves, because see, as Thangaraj mentioned, that in future there would be another epidemic or pandemic. We should be prepared and mentally ready to how to protect ourselves. So this kind of awareness was not there, and this kind of what all things pathogens like malaria, majority of this problem, malaria, dengue, all these infectious disease, we are actually mostly victim because we're casual and we are not really aware and protect ourselves that much. So at the community level, if that can be implemented, I think that is a big achievement if we can do it. Social awareness is something very important, along with the research and whatever the other things that we are talking about. Support system.
Thank you, sir. On that note, we wrap up this webinar. Thank you very much, all our speakers, for joining us today, giving us your valuable time. I'm sure our audience has benefited a lot. A quick note for our audience: when the webinar ends, a survey will pop up on your screen. We'll be grateful if you can fill in the survey quickly. It will help us improve the quality of our future webinars. Thanks again, all our speakers and audience, for joining us today. We hope you have a safe weekend. The addresses of DBT website are displayed on the screen. If you wish to know more about the initiatives of DBT in mitigating this pandemic. Thanks again.